Hello, everyone. I pray we are all enjoying the preparations for this Advent season. I will be presenting Variola Virus Infographic 1 and 2, commonly known as smallpox. I hope it's informative. I'm sure you are all aware that the infographics are full of information, and all the density of information is hard to present. I was challenged, and this is my best try, so I'm attempting to present the infographics on PowerPoint with the morph features. I hope it's presentable. Here we go. Variola virus, smallpox, is thought to have been causing disease in humans for at least 3,000 years, and lesions resembling smallpox have been found on the faces of Egyptian mummies. On the left side, you see the pox marks left by a suspected infected individual about 3,000 years ago. It was declared eradicated in 1980, as the last case was known to be in Somalia in 1977. The classification of variola virus comes from the family of Poxiviridae, the subfamily Protopoxiviridae, the genus Orthopoxivirus, the species Variola virus, and the Baltimore classification double-stranded DNA. Other members of the Orthopoxivirus genus include Vaccinia virus, and I will show you here. Here's the Vaccinia virus, and the monkeypox virus, oops, up here, and the cowpox virus, which is right there. All these stem from one common ancestor, the Protopoxiviridae, and they come from the Poxiviridae family. Smallpox is highly infectious and exists in two variants. The variola major, with a fatality rate of 30 to 40%, is the focus of the infographics. However, the second variola minor, with a fatality rate of around 1%, is less fatal and results in a minor fever, minor rash. The two differ by about 2% in their base sequence. The anatomy and structure of variola virus is quite unique. The shape is considered brick-shaped or oval. The size is approximately 300 to 350 nanometers in length and 200 to 250 nanometers in width, and it is considered a large virus, comparatively speaking. It is a complex envelope structure with two forms of infectious virions, which we will discuss in more detail later. There is one called the intercellular mature virion, IMV, which has a single membrane, and the extracellular envelope, which has an additional outer membrane that aids in the host spread through the blood. So the intercellular IMV has one membrane. The extracellular envelope virion has two membranes. One is an envelope. The capsid symmetry is complex symmetry. It's neither purely helical nor icosahedral, which is typical of poxviruses. The genome is linear double-stranded DNA, covalently closed hairpin termini, and no free 3' or 5' ends. The size of the genome is approximately 186 kilobase pairs, so it's quite a large virus, and it is, as you see, pretty unique in its replication and relentless in its infectious properties. Its genome is also very interesting. The variola virus genome encodes for about 200 genes, though only 150 are functional in variola major. The gene organization is conserved, it has a central conserved region. It encodes essential genes for replication, transcription, and virion structure. The terminal variable regions encode host interaction and virulence factors. The replication, it also encodes for its own replication enzymes. It encodes for its own DNA-dependent DNA polymerase, transcription factors, capping and polyadenylation enzymes, allowing replication entirely in the cytoplasm. Unlike most DNA viruses, it has everything it needs except the energy and the cell space. And because it encodes for everything it needs, the genome contributes to its relentless infection, as we will see further. The replication process is quite unique, and the strategy is just incredible. Because there are two types of virions, the envelope virion, which is the extracellular version or virion, the intercellular, which only has one membrane, which stays inside the cell. They both attach to the cell surface differently, as they have different receptor sites, and they also enter differently. So entry occurs by macropinocytosis for the IMV, and direct membrane fusion, depending on the virion, for the EEV. So this one, because it has the envelope, can directly fuse to the lipid bilayer, and this one needs pinocytosis. Once inside, entry occurs, once inside the cytoplasmic viral factories, the virus carries a complete transcription system, allowing early mRNA synthesis immediately after entry. Uncoding occurs. All the necessary enzymes start their replication process immediately. DNA replication begins once early proteins are synthesized using the viral DNA polymerase. Intermediate and late expression, intermediate genes trigger late gene expressions, producing structural proteins and enzymes needed for assembly. So once it enters, it uncodes, releasing the genome and everything it needs to replicate and transcribe that genome. Early proteins, late proteins, and the assembly begins. Assembly occurs in the cytoplasmic factories, where crescent-shaped membranes form mature virions, which mature into IMVs. So here we see the replication factory and the production of the intercellular virion. Now, this is where I feel it's very unique in the release. So this intercellular virion will morph into the mature intercellular virion, or it will go to the Golgi apparatus to be enveloped and exit through exocytosis. And part of the exocytosis with the envelope from the Golgi apparatus aids in the evading of the immune system. Or it will stay in the cell and replicate until the cell lyses. And these do not have the envelope, but as soon as they lyse, they will go on to host the cells, the other cells. So the release depends on the type, if it has an envelope or not, and also will determine the host range. And this is why it's relentless. I'm assuming that these will go to the ends of the limbs, and these will infect the oropharyngeal, which we will discuss later. But I thought it was quite unique that it has two mechanisms of exit, making it very relentless. I think you would agree. Tropism, because of the two unique virion types, the tropism is pretty relentless, as I've mentioned before. It affects cells, tissues. Host range, variola virus is strictly human specific. There are no animal reservoirs known, so its life cycle is strictly human. The primary target cells, it targets the epithelial cells of the respiratory mucus layers. So all or the mouth, the throat, lymphoid tissues, where it replicates tremendously, endothelial cells and dermal epithelial cells, as you see in this picture, how it affects the hands, the feet, and the face, and the legs and arms, as you will see later. Tissue tropism is systemic infection. It leads to replication in reticuloendothelial tissues like spleen, liver, bone marrow, and subsequent infection of dermal and mucosal epithelial, causing the characteristic rash, which is so relentless. Now to the second part of the infographic, the variola virus part 2. Virus part 2, smallpox. One of the most devastating diseases known to humanity, caused millions of deaths before it was declared eradicated in 1980. The global burden before eradication caused 300 to 500 million deaths in the 20th century alone. Typical case fatality rate was 30% for variola major and 1% for variola minor. It was endemic for a millennia. The earliest evidence, as mentioned before, was from Egyptian mummies in the 3rd century. It affected every inhabited continent before eradication. The World Health Organization, the WHO, launched a global eradication campaign in 1967, and with a lot of effort on the ground and all around, it was declared eradicated in 1980, as the last case occurred in Somalia in 1977. Transmission occurs mostly airborne through respiratory droplets, secretions of infectious viral particles that are released from the sloughing of the oropharyngeal lesions. Or transmission also occurs through direct contact with the lesions from the onset of lesions until all the crust has sloughed. Or it also transmits through contaminated clothing or bed linens. As we saw in the beginning of the slide presentation, the U.S. Army was giving the Native Americans the infected bed linen to infect the Native American tribes and kill them off, as they were considered, quote unquote, a problem. Infection tends to spread slowly and mainly among close contacts. Infection is most communicable for the first 7 to 10 days after the rash appears. And in this diagram here, we see that, yes, it's exclusively human host, but the rate of spread is called centrifugal spread, as it spreads in a circular fashion throughout the body, face, arms and hands, legs and feet. And so this is one way that healthcare professionals were trained to identify the difference between smallpox and chickenpox. Genesis is high level. Entry, as we discussed earlier, is via respiratory mucosa. The variola virus replicates in the lymphoid tissues. Then it spreads viremia to the blood, dissemination to the skin through the blood, and then lesions develop. The immune response contributes to the system. Immune response contributes to the symptoms, the high fever and the extreme pain, and no doubt the inflammation response. Other organs are seldom clinically involved, except occasionally the central nervous system with encephalitis. So on occasion, rarely though, the nervous system is inflamed. Symptoms and clinical course occur in the following way. Number 1, incubation is 7 to 17 days, then followed by prodromal fever, then centrifugal rash, as we saw in the last slide. The rash has different timelines. So as you see in this image here of a child who's been infected, the rash on the 3rd day appears, and then on the 5th day, it starts to inflame or pustulate, and then eventually it'll scab. I found most of the pictures pretty disturbing, so I didn't include too many, and it just for the dignity of the person, but I thought this one was acceptable. I hope you all don't. I hope it's not as disturbing. Then number 4 leaves pitted scars, and number 5 severe variants include hemorrhagic and flat smallpox. So those are some of the symptoms, and let's go on to the treatments that are available. Treatment options for smallpox is mostly supportive care, isolation to prevent the spread. There are modern antivirals, and there is a vaccine. If you notice, the vaccine is delivered with this interesting needle that has sort of these 2 little spikes. These are to cause some kind of skin lesion. So about 10 times the skin is scratched, and the vaccine is delivered to induce an immune response and deliver the vaccinia, the virus that is less virulent and cause an immune system. Or I'm sorry, cause an immune response, pardon. There are no known applications for variola. It is strictly regulated, so it has no role in agriculture, food science, or engineering. The remaining variola virus stock has been restricted to the WHO collaborating centers on smallpox and other poxvirus infections at the CDC and the Russian State Research Center of Virology and Biotechnology. This concludes the information on both Infographic 1 and 2. I hope it was informative. I will be happy to answer any questions as best as I can. Wishing everyone a beautiful Thanksgiving and a wonderful Advent season. So happy Thanksgiving and Merry Christmas. The last slide includes the references. God bless.